Hey there, how are you doing?
Good. I don't know why my AirPods are not working. Okay. So I actually figured out what happened with my OneNote.
Yep.
Um, so remember I told you that my OneNote was like, or my Office 365 was through my school.
Mm-hmm.
So I apparently like yeah, when you graduate, they kind of cancel the membership.
Yeah.
So I have to get like an actual
Uh-huh.
non-student Office 365 membership or something, like a yearly membership.
Mm-hmm. You can
So
still
what I'm,
access it online though, right?
what's that?
You could still access it online, right?
Oh yeah, but like for example, if I want to use it for my iPad and stuff, like to take notes for other things,
Mm-hmm.
I need to have like a subscription to Office 365. I
Hmm.
guess I don't know.
Okay.
Yeah.
Because I don't think I have a subscription to that. But,
No, do you actually take like when you take the notes or when you actually write down, you have an iPad, right?
oh, not on an iPad. No, I use Windows.
Oh, you have a Surface or something like a tablet?
Oh, yeah, I have the Surface.
Oh, but then that comes with the OneNote app.
Yeah, probably. Apple always charges for stuff. Um,
Yeah.
but I also like. Okay. Um, But yeah, we can use the online version now,
Oh
and
yeah, yeah, yeah. That's fine.
and you're able to edit it online, I believe, right?
Yeah, but how would I write if I don't have a pencil?
Oh, I mean, like if you use it online on your iPad, if you're able to edit it,
Oh,
you can perhaps do that. Yeah,
okay.
and like worst case, well, I mean, like this isn't for everyone, but I like pirate all my stuff, so that's whatever worst case. But it seems like yeah, you could still use it for the most part.
Okay.
Um, all right. So, what would you like to work on today?
Can we continue with the chemistry physics?
Okay, that section bank, of course.
The yeah, at least for a little bit, and then I want to touch a little bit on if we can find some questions for um, amino acids and enzyme reactions for biochem.
Uh huh. And like any particular question regarding that?
Well, I don't think we can filter that in double AMC, but maybe we can do it through the filtering system of UWorld. So I
Yeah.
can just open some questions from there, and then I can share my screen with you, or I can give
Yeah.
you my login information, whatever you want.
uh, but yeah. Within that, what types of questions? I guess. Um, because all you really need to know are just the basic ones, the acidic ones.
But for example, like questions about like isoelectric points
Oh
and
yeah.
like enzyme reactions and stuff
Yeah,
like
yeah.
that.
So like isoelectric point is just basically you alter like the pH for it
Mm-hmm.
to reach. So the isoelectric point is the pH at which the amino acid is neutral. Like a Zwitter ion.
Mm-hmm.
And um like isoelectric focusing, when they use that gel, they they could have like a number of like peptides, and they see how and they and it's across a pH gradient, and they see where it moves to, and that will be the isoelectric point. The isoelectric point, just think of it as, I mean, like iso meaning like the same, and it's just the pH at which it will
The
be.
amino acid is neutral.
Yeah. So generally speaking, if it's a neutral amino acid, the isoelectric point will be around like 7. And sorry, one sec. Hello. Yeah. Yeah. Are you here? Okay. Okay. Yes. Yes, I will. All right. Thank you. Bye. Sorry, that was a plumber.
No, it's okay.
Okay, so yeah, so that's isoelectric point stuff, and let's see, and
So why do they say that you have to memorize all the isoelectric points for?
because they say
I mean,
that
like,
about everything, you don't need to memorize any
but
of
all
that
the
stuff.
amino acids that you need to memorize.
You only need
also.
to memorize, the basic ones and the acidic ones.
Okay, but yeah, which I already know them, but like,
Mm-hmm.
do you need to also know like the you need to go into the structure and all of their
Uh-huh.
characteristics, or just know like which are the basics, which are the acidic, and that's it.
So like, all right, if you wanted to calculate the isoelectric point, right? Um, so okay, every amino acid has, a C terminal and an N terminal, right?
Wait,
Yeah, sure.
okay. Oh well, just remember to whenever we start going over the questions to
Yeah.
record it so I can go over it later. I
Okay.
just remember, sorry.
All right. Okay. I'm recording it using a different
Oh, that's fine.
app
Yeah, don't worry.
thing, so I can get like the full screen, because when I tried recording through Zoom last time, when we did the shared screen, like it would only show this shared screen, so it was just like irrelevant stuff from my notebook. But this time I have
got
like
it.
everything, like yeah. But yes. So okay. So the amino term. So the N terminal, the C terminal. So you know what I mean when I say that, right?
Yeah,
Like, what is at the N terminal?
an amino group.
Good. And then carboxylic acid group on the other one, right? And the pKa of the carboxylic group is like roughly around like 2 something. And for the amine group, it's like around like what, like 9?
Mm-hmm.
So isoelectric point, you just calculate the average of those two. Now, if it was, so yeah, you'd get something like 5.5, right, or like 6. So if it's a basic one, there's gonna be a side group or side chain, or yeah, side group that will be more basic. So it'll have a pKa that's gonna be higher. So you just take the two highest pKa's and average that.
Wait.
Sure.
So if it is a basic amino acid, then it's going to have a higher pKa. And what you said you take that pKa and you take the 2 pKa's?
Yeah, you're always going to take an average of pKa's. But if it is a basic one, you're going to take the higher average of the higher pKa's. And conversely, if it's a acidic amino acid, you're going to take the average of the lower pKa's. And so the isoelectric point for a neutral amino acid will be around 5.5. For an acidic one, it'll be lower than that. And for a basic one, it'll be higher than that. And yeah, that's all to it. An actual value, you can still, calculate that, but as long as you are able to see, conceptually how the isoelectric point, will shift, towards something greater than, 7 for basic ones and less than 7 for acidic ones.
Mm-hmm.
Yeah, because
I think we should do some questions about that.
Okay.
So I can see exactly what.
Sure. All right, so let's get to it. I mean, we can do some of the BB section bank. I'm sure there's tons of questions
for sure.
in that also. So Okay, so all right, so yeah, which one do you want to do? You want to do some BB then?
Mm-hmm.
Okay.
Yeah, let's start with the positive side. Let's start with the easier part.
Okay.
Uh,
So all right, I'm
Let
gonna
me
open up
open them.
the section bank for BB.
Why can I find them? It says question path. That's weird. Oh. Okay.
Okay.
Oh, wait. It's like logging me out. Oh my God.
It's okay.
There's so many things that I have open right now in my little laptop.
Yeah.
Section bank. All right, I have the first question. The passage is about blood flow to the brain, blah, 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 blah.
Mm-hmm.
Okay.
Okay, so all right, so this gives us an opportunity to go over the passage.
Okay.
Here, one sec. Okay, so
Wait, did you just read that passage?
no, I just like
Oh, I was
moved.
reading the passage and I was like, oh my God, did you just like read the whole thing?
No, but all right, now we're gonna read it together. So,
Okay.
well, first of all, I look at the question, and it says what type of enzyme is likely to suppress CBC. So now I'm gonna look for CBC in the passage, and I
You
see it.
can't do Control F when you're taking your test, can you?
No, you can't. So
Yeah.
yeah, you have to get used to being able to find things really quickly in the passage, and I know that's easier said than done.
No, but you get used to it. I mean, I do the same thing in cars. Like, you have to
Yeah,
constantly do it.
yeah, and
Okay.
I've been reading passages like this for like my entire life, so it's I see it. It's at the end of the second
Yeah.
paragraph. So it's talking about a crossbridge cycle for actin and myosin, and like you remember that from whatever content review. Like that's when a muscle contracts. All right. So they're saying that phosphorylation of something is required to make this happen. So what type of enzyme causes? Oh wait. It says to suppress it. So they're saying that
Yeah.
So like okay, when it comes to highlighting, I would highlight phosphorylation.
On the passage, so phosphorylation of LC20 is required to activate the myosin head.
Yeah, just highlight phosphorylation.
Okay.
And like the shortcuts like Alt H and stuff.
Shortcuts for, I'm sorry?
Like the keyboard shortcuts.
Oh, no. I actually click highlight.
Oh
and
yeah,
stuff.
yeah. Use the shortcuts because they're shortcuts.
You're
So
a keyboard person.
yeah.
You know all the shortcuts for keyboards and stuff. Like,
Mm-hmm.
so what is the shortcut for that one?
Oh, it's hold Alt and then you hit H. So basically, when I'm reading a passage, my hand is already over the keys Alt and H, and I just double click on phosphorylation, and it highlights it, not like highlight it, but this like the mouse, the normal highlighting that you can do with anything. Then I hit Alt H and it highlights.
Okay, let me try that.
Mm-hmm.
Okay, got it.
All right, and also if you hold Alt and hit S, it does a strike through. If you wanted to know that.
Oh, okay. So you double click and then you do Alt S for strike through.
Yeah.
Oh no.
So yeah.
Alt S.
Yeah. So yeah, highlight phosphorylation and then highlight CBC.
Okay, but wait, the Alt S doesn't really work for highlight for strike through.
All right. We don't have to worry about that.
Okay.
All
right. So phosphorylation and then CBC.
Yeah. So
okay.
All we need to. So that just tells us phosphorylation leads to CBC.
Mm-hmm.
And they're asking what type of enzyme would make CBC not work.
Mm-hmm.
So
it's going to be an enzyme that removes or inhibits phosphorylation.
Exactly.
So that's positive?
Yep.
Okay, so we didn't even have to read the passage.
Yeah.
Question.
Yes.
Do you always do this? Like, do you always first look at the question before reading the passage?
Yeah. Because, at most there's 5 to 6 questions per passage, and like most of the time you can get through those. That's a very small amount of passages, because these passages are from like research papers,
Mm-hmm.
This one, it says this is from a paper that was published in Nature, so
Mm-hmm.
I don't know if you've done any like research like in labs and stuff, but so like reading all those papers, it's a hell of a lot. But so it doesn't really make any. There isn't really a point to like completely know every single tiny bit of what everything means, because we're not really going to use this for any other reason than answering just the questions that they give us. Just take it one question at a time, instead of one passage at a time, a question at a time.
Okay.
And if it calls for us to read it, then we go to the passage. So all right, so this one made perfect sense to you, right?
Yeah.
Okay, so now this one is going to be all right. So they're saying which statement regarding VSM function and cytoskeletal dynamics is best supported by the data in Table 1. So now we're going to basically look at Table 1, but I would also look for VSM.
Mm-hmm.
And I see it. Do you see it?
Yeah, so second to last paragraph, dynamic reorganization of the actin. Okay.
Yes. So basically, I would say, so I would highlight reorganization.
Mm-hmm.
And I'd highlight cytoskeleton.
Cytoskeleton. Mm-hmm.
And then I'd highlight VSM, contraction, and I'd highlight relaxation. I wouldn't even highlight the and part. Like, you want to highlight as little as possible, just so you get to see like cytoskeleton reorganization causes VSM contraction or relaxation. If you because if you highlight the entire sentence, the more you highlight, basically, the less highlighting is worth it, right? Because highlighting is meant to, emphasize certain aspects of it. So if you highlight everything, it's not going to emphasize it, right?
Okay.
So there. Okay, now we look at the table, and we see different things about pressure. Okay, so for the table, since right now it's not like completely obvious, I'm going to like kind of look at the paragraph above it,
Okay.
to give it some context. So it seems like it says changes that occur in VSM during the MR, which is the myogenic response. That's up on the first paragraph.
Okay.
So I would highlight there, MR.
So,
Mm-hmm.
for example, like, don't you think that while you, and this is not like, I'm not saying it's wrong, I'm just saying
Sure.
like in this case, when you're saying, so, okay, I look at the table and I'm like, I have no idea what the table is talking about. So let me just get a little bit of context. And then I read the paragraph above it, which says, so the timing changes that occur in BSM during the MR. And I was like, and then I'll be like, what is MR? So I have to go up and read what MR is. So basically I'm like reading the passage, even if I don't want to, or like even if I didn't do it initially.
Yeah, because it shows you which things to focus on. If you read it without the questions, you're going to not know what to focus on.
Right, because you don't know what they're going to be asking you.
Yeah. So now I know that the MR basically causes, vasoconstriction, right?
Mm-hmm.
And so now the table, is telling me like arterial diameter. So they're basically changing, Okay, so anyway. So do you see how it says MR? So I would highlight MR, and I would highlight cerebral and vessels. And then, yeah.
And vessels.
Mm-hmm.
Where are you looking for vessels? Where are you looking at vessels?
Top paragraph.
Oh, you're talking about the top paragraph.
Mm-hmm.
Okay, so blood flow to the brain is regulated by changes in the arterial vessel diameter in response to intravascular pressure. Okay, so MR, cerebral, and vessels.
Yeah, and then constriction.
Mm-hmm.
And then dilate.
Constriction and dilate. Okay.
So basically, what that tells me is that the MR is responsible for vasoconstriction or vasodilation in the blood vessels of your brain.
Okay. I was just reading. Sorry. Yeah.
Yeah. So okay. So now I am going to take a look at. I mean, I kind of have a good idea of the table, but I'm going to, look at the paragraph right above the table. And it's basically just saying, in the experiment, they're trying to change, VSM, vascular smooth muscle, right? AKA, the blood vessels, right? They're trying to examine the changes in it, right? Whether it dilates, whether it constricts, right? And they're basically just measuring arterial diameter. So, okay, let me ask you this. Like, if the arterial diameter... So what happens to the diameter of the arteries when vasoconstriction happens?
It decreases.
Correct. Good.
Mm-hmm.
Okay, so they're measuring that. They're measuring the amounts of phosphorylated LC20.
Mm-hmm.
And then,
how is phosphorylated LC20? So it is required to activate the myosin head, which will cause muscle contraction. Okay, so that will be an indication of arterial contraction?
What?
No.
Sorry. Wait. Causing what?
No, because so they're measuring arterial diameter, right? They're measuring phosphorylated LC20. No, just keep going. Don't worry. Keep going.
It's okay. I'll write some stuff in the notebook as to how I would like process this. So I'm going to write, but there's surely going to be a delay.
Yeah, don't worry. Let me actually get something to write with. Okay.
Yeah, another way of going about this is looking at the variables in that table and then looking for that in the passage.
So, for example, looking at.
Do you have my
okay, yeah.
OneNote?
Diameter.
Do you have my OneNote open?
Yes.
On session 2, does it show you what I wrote?
Yeah.
Okay, so this is like a shorthand. So I wrote MR is proportional to VSM, and that can either constrict or dilate. And LC20, when it is phosphorylated, I drew that as that P in the circle, causes the CBC to happen. And now I'm going to look at calconin. So now, calconin, and when that is phosphorylated, it says
It reduces its inhibition of the actin activated myosin MGATP.
Okay, so that basically means that it's going to when it's phosphorylated, it lowers its inhibition of the ATPase. Sorry. Hello? Oh, okay. Okay. Call me when you're here. Bye. Sorry, I have to have a plumber here and fix stuff.
Oh no, it's okay.
Okay.
So phosphorylation of calconin reduces inhibition of actin activated myosin.
Yeah. So basically, like, it removes the ATP. I suppose. So if it's phosphorylated, it'll cause more of the ATP to like connect essentially. That's all. I mean, like that's all we need to know for now. But it should kind of like click with some of the things that you did in content review, maybe. If not, it's still fine.
No, I know this part. Like I'm familiar with muscle contraction, but I'm trying to think. So if I think I find confusing the part that it says it reduces its inhibition. Okay, so phosphorylation of calconin reduces its inhibition of what inhibition? Its inhibition of what?
Its inhibition.
Arterial diameter and the amount of phosphorylated LC20.
So anytime you see inhibition, it will do that to an enzyme. So then you look for an enzyme, and they're going to be, ending in ASE. So the enzyme here is MG ATPase.
Oh, MGATPase. Okay. Okay.
Okay. So yeah, and then let's see. All right, G Actin were determined in response to a three. So G Actin is just a type of actin. Like, so I'm looking at that like passage above that one that we're looking at, which is basically the third passage down.
Yeah.
And maybe I'd just highlight G-actin, polymerize, F-actin.
G-actin, polymerize, okay.
And then there, you know what? Might as well add this. They haven't asked it yet, but it just says depolymerization can be induced by the drug, like LAT-B, right?
B, okay.
So I would, draw like an arrow going in the opposite direction with, LAT-B.
Okay.
Yeah. So, what do you think the MG ATPase does?
So it actually hydrolyzes. Well, it will break down ATP.
It'll get. So if something gets phosphorylated, right, it's going to like dephosphorylate it.
Right, so it's going to dephosphorylate ATP.
Yeah, it's basically the answer of that thing that we had in the question before. Like it's a phosphatase.
Right, so basically it turns ATP into ADP.
I think so. Yeah.
No?
It takes away a phosphate group essentially.
Okay.
Yeah, and so I basically wrote that in the notebook, and that's why. By the way, when people die and they get rigor mortis.
It's because there's no more ATP available to allow the myosin head to detach from the...
Exactly. Good. Yeah. So
yeah.
You know all this content. Okay. So now let's just look at basically what happens when we alter the the interior pressure, right? The internal pressure of whatever a brain blood vessel. So, okay, if we increase the pressure, it seems like and then I'll write this also. So, whatever, diameter. As we increase the pressure, the diameter decreases, right?
Right.
Yeah. So I'd say, it's inversely proportional to the pressure.
Okay.
All right. Now, phosphorylated LC20, as we increase the pressure, it increases. I mean, it reaches like somewhat of a stable point. But yeah, it increases. So then I'd write whatever. Phosphorylated Oh, where's my drawing go? Okay. Okay. And then phosphorylated calconin, also proportional to pressure. And then the amount of G-actin. Indicate data are normalized using a 10. Okay. I mean, yeah, that seems
What do you...
Yeah?
Oh, what is that? So indicate that data are normalized. What is that important?
Yeah, so since everything else has units, right? Or is a percentage. And G-actin is the number of it. They're just trying to tell you that the numbers like 1 and 0.3 or 0.3 and 0.1 and stuff are they're not like how many molecules are there. They're just compared to the standard or the control or whatever, which is 1. So basically what it's telling, so it's like let's say that 1 is like the normal amount. It's really like going to be like, like millions or billions of these molecules, right? So
I knew what normalization was. I know they do it a lot with protein when like they're trying to measure protein quantity, right?
yeah, when you normalize something, essentially what you're doing is you're setting like a basic kind of standard, and you compare it to that standard instead of having like an exact amount. So, yeah.
So, for example, what they did here for G-actin was that at 10 millimeters of mercury of internal pressure,
Yep.
the amount of G-actin was 1.
Yeah.
One what? 1
exactly.
unit?
So there's no. So that's why they had to normalize it, because we don't know how many. Like, they're not going to give us like the exact number of molecules of G-actin. That's going to be hard to figure out. So they just use that as that standard to say this is 1. Think of it as 100%.
Okay, so then for 80 it would be 30%, and then for 120 will be 10%?
Yeah.
Okay.
So like,
Got
yeah,
it.
we're just looking at changes basically, right?
Mm-hmm.
So yes. So basically, like, whatever number, or rather, I would write concentration of G-actin, and it goes down, right? So that's also inversely proportional to proportion.
Mm-hmm.
So basically, we just went over the entire passage, more so than we need to for this question, but now we can look at that question.
Okay.
So, which statement regarding VSM function and cytoskeleton dynamics is best supported by the data in Table 1? Okay, so we'll go through, each of them. So, vasoconstriction is associated with an increase in ratio of F-actin to G-actin. Now, what do you think about that?
Well then, so if vasoconstriction is inversely proportional to the pressure, but the amount of G-actin, it's also inversely proportional
Well,
to the pressure.
Well, A isn't saying anything about pressure yet. It's just saying, is it true? So, So G-actin turning into F-actin happens through what? Like polymerization, right?
Okay, yeah.
So, and
But
yeah.
since it said vasoconstriction, that's why I looked at the arterial diameter, right?
Oh yeah, but they're just choice A isn't saying anything about that. All it's saying is that well, okay, we can look at it that way. So vasoconstriction occurs as we increase the pressure, right?
Yes.
Because that diameter decreases.
Mm-hmm.
Okay, and then the G-actin amount decreases as well, right?
Yes.
All right, and it decreases because it turns into F-actin, right?
Mm-hmm.
So A would be true, wouldn't it?
Yes.
Okay. Now, of course, we're not going to leave it there. Let's look at B.
Oh my God, no. So it's A.
So
okay.
B is saying F-actin levels decrease as phosphorylation of LC20 increases. So
that is F-actin levels decrease. Oh no. It's actually F-actin levels increase.
correct.
Yeah, because if G-actin is decreasing with phosphorylated LC20, then F-actin needs to increase.
Mm-hmm.
Okay.
So B's out. And then let's see. Vasodilation does not affect phosphorylation levels of calconin. Here, I'll be right back. I have to just talk to this plumber. Okay.
Okay.
All right, let's see. So, vasodilation does not affect phosphorylation levels of calconin. So, what is that,
No, it actually affects. I mean, it does affect that at some point because if you look from 120 to if you actually compare the arterial diameter, it stays at 18, but then it goes down to 7.
Yeah.
So it does affect it at some point.
Yeah, it goes from 7 to 18. After that point, it doesn't, go any further.
Right.
So, but it does certainly affect it.
Okay.
Yeah. And by the way, it's probably, reached its saturation point at 18. That's probably why. That's the same thing with LC20. It goes to 50 to 52, not that much of an increase because the enzyme's probably saturated at that point. It's reached its Vmax, right? It's the maximum amount that it can take in. So, but yes, it certainly affects it. So C is out. And then it says arterial diameter reduction, aka vasoconstriction, is always dependent upon increased calconin phosphorylation. And that's where you see that, calconin phosphorylation is 18 and stays at 18, yet the diameter changes.
Decreases.
So it's not always dependent on it, right? Yeah, so the answer will be A.
Okay.
So.
Wow.
Yeah.
How can you do this? And I mean, the interpretation seems you just have to get the relationship between the variables pretty much.
Exactly.
But then you have a minute and what is it? A minute and 18 seconds for that.
Yeah, I mean, of course, on the actual test, once you see, we did get the answer immediately, but do you see how going through the choices is the way to read it? Whereas if you just read it, people would be writing MR equals myogenic response, and and from, my OneNote, you get to see exactly how I'm writing it and just how things are proportional. Yeah, if you can think in terms of just this is proportional to this, because if you could think in those terms, you're, that's it's a wrap. You got it. So
Okay.
So let's look at the next one.
Mm-hmm.
So compared to controlled RMCA, rat middle cerebral arteries, basically, just VSM. Also, like, you see how I'm putting like all these things in like the same, like I'm saying, because they want to put a bunch of different words in your head to psych you out.
Yeah.
And again, like I'm saying, like people who usually take it, they're gonna write, oh, it's RMCA. It's rat middle cerebral arteries. It's not the ones that are not in the middle, but it's just VSM, which is just VSM, which is vascular smooth muscle, which is just blood vessels.
Mm-hmm.
Okay, so basically, compared to control, held at an internal pressure of 120. Yeah, hold on. Yeah. Okay, so application of lat B. So like sometimes you get a sense of like knowing.
Did you know that this was going to be on the passage since you emphasized on lat B?
I mean, like, I technically did this before, but when you read it, like, I just like saw polymerization, and then something that does the opposite by a drug, and most of these passages are going to involve some question about that. So
okay.
So anyway, when they do that, right, it's going to stop the polymerization, which would mean what?
They would, hold on, research.
In my notes, do you see what I wrote with G-actin and F-actin?
Yeah, so you said that G-actin polymerizes into F-actin, and then lat B does the opposite thing.
Exactly.
So depolymerizes, right? And then, okay, so that happens whenever there is VSM, right?
Oh, all this stuff is happening to the VSM. But basically all you need to know, though, is that, like, what's going to happen to the... I mean, if polymerization causes there to be more F-actin...
There is going to be VSM, or at least that's what I read on the passage, that on the paragraph it says VSM contraction and relaxation.
Yeah, just look at the part that says polymerization causes G-actin to turn into F-actin.
Mm-hmm.
So depolymerization would cause F-actin to turn into G-actin.
Mm-hmm.
So the amount of... Tell me what happens to the amount of G-actin or F-actin.
Right, so whenever there is lat B, the amount of G-actin is going to increase.
And the amount of F-actin?
Is going to decrease.
Yep.
Oh my god, I see.
Yeah.
Okay, because I haven't read the question. So compared to control RMCA held at an internal pressure of 120.
Yeah, we don't even need to know about the 120, by the way, but...
Yeah, why are they giving that 120 though?
That's what they do. That's how they... Like, they give you all this information because they want you to think the 120 is important. And they want you to look at the table and look at where it says 120, and they want you to look at G-actin and see that it says there's less of it, and then they want you to see, not choose the answer that says that there's less of F-actin.
Oh, wow, that's mean.
I mean, like, yeah, that's why, I'm saying, like, like if you can get to the point where you can separate the useful information from the useless information, you're good. And so the way that I wrote like things out, that kind of shorthand is like how you want to like write it out. You don't want to write any sentences. You don't want to write, what VSM is, because I guarantee, because 270 people take this test like a year, right? And they're smart. They're like the smartest. Like pre-meds are like the smartest people compared to like other groups, and so this is like a hard test, and they have to make it hard because they need to like they can't let everyone in, right? So they're going to, and another thing is pre-meds are the type that take thorough notes about everything, right? And yeah, going about, this test you can't go about that way because it's never gonna work.
That's what you see, or like it has happened to many of my friends. Like they have like really good GPAs, or like they're really good like in their classes.
Yeah.
But then they take this test, and whatever they get, it's like not proportional to. Oh, sorry, my phone.
Yeah.
Not proportional to what they did in college, like.
Yeah, and again, remember that's why math majors and physics majors, like score the highest.
Yeah.
Like even though this is about, like biology, right? Because
It's more rational and logic.
Yeah.
Stuff.
And in physics, basically, where we are just looking for, it's very reductionist. We're reducing everything down. But yeah, so that's the answer right there, right?
Okay.
Okay. And by the way, so they start off by saying compared to a control, right? So it's not going to be, we're not going to be using anything in table 1. But yeah, that's also why, I say like I want people to know like why the wrong answers are wrong, because the wrong answers are designed to be possibly right if you think about it in a way. They're like kind of like leading you on to like the wrong like down the wrong path.
Right.
Okay. But yeah.
They sound, what you mean is like they sound right. So for example, I see a lot of that in cars though, like the most elaborated and the ones that sound prettier are usually the wrong ones.
Oh
like,
yeah.
Whenever you're like, oh my god, this has to be right, but then you don't really know what it means, and then you click it and it's wrong.
Yeah, we could do like a car section as well. Or
like I would love to
passages.
do that Picasso passage that everybody talked about.
Yeah, it's like, it's really not as bad as people say, but but again, so like everything that I'm trying to do, when I wrote the cars guide for my other student, it was just me like reverse engineering the way that I think. And so okay, but all right, let's move on to the next one. According to the information in the passage, which statement best describes the function of the MR in response to stimulation of the sympathetic nervous system? So basically, what does it do? So,
well, it says here that the MR allows cerebral resistance.
So in the brain,
partial
right?
constriction or dilate. So basically, it alters blood flow.
So, but in the brain.
Right.
So A is talking about aorta. So that's out.
Oh, okay.
Now.
Moderates blood flow to the brain under high pressure.
Yeah. And it says, in response to intravascular pressure, right? In the first sentence.
Oh, in response to intravascular pressure. Okay.
Yeah. That's why it's a response, right?
Right. So does this high pressure where they're referring to is what, like not intravascular, they're referring to actually
intravascular.
External pressure?
Intravascular. Because think about it. There's no there's no other pressure in our bodies really that we can measure. Right? It's just like blood pressure, right?
Mm-hmm.
Yeah.
Okay, so that can be an option.
Yes.
Because
And
C says enable cerebral resistant arteries to locally vasodilate.
Okay.
I mean, that can be an...
So what's the difference between that and B?
Yeah, they sound pretty much... Well, that B is saying that it moderates it. Basically, it allows for either constriction or dilation.
Correct.
And then C says that it only enables dilation.
Exactly. So so C's out, right?
C's out, yeah.
Because any type of statement that is like absolute means that it's easily proven wrong, right?
Okay.
So C is out, and then D.
Forget it because it says abdominal cavity.
Yeah, which by the way, that's what happens in the parasympathetic nervous response.
Right.
Yeah.
But then really the stimulation of the sympathetic nervous system doesn't really matter.
Yeah, it doesn't. I just wanted to add that bit.
Oh.
Yeah. So
blood flow to the brain is regulated by changes in arterial beats. I just want to know how did you actually got so quick the fact that it was only to the brain, because then I would be thinking like.
Oh, we did that from the beginning, remember? Like
Yeah. Well, like.
That it's just we're talking about it in the brain.
Mhm. Yeah, no, it says it there. It's true. Okay.
Yeah.
So it's B, moderate blood flow to the brain under high pressure.
Mm-hmm.
Okay.
Yep. That makes sense?
Yes.
Okay. Okay, then the last one, it seems, is... Oh, no, wait. There's two more. Okay. The polymerization of which structural component... Okay. What do you think?
Oh, this is easy.
Yeah. This is, like, pretty much kind of, like, whatever.
Content, yeah.
Mm-hmm.
Well, I think it's microtubules because it's F-actin and G-actin.
What's actin made of?
Is it tubulin? No.
Well, microtubules are made of tubulin, that is true, but... Okay, so... Yeah, check out, like, the Anki stuff, because I talk about the differences between these three. But, yes. Myosin is made of microtubules, and actin is made of microfilaments.
Mm-hmm.
Yeah.
Okay, and they're talking about F-actin and G-actin, so it's microfilament.
Yep.
Okay. This is like pretty. This is just like either you know it or you don't know it.
Wait, what's that? So
this question is more like either you know it or you don't know it. Like.
Yeah, but I would say like as far as content is concerned, this is definitely something that you have to know.
Yeah.
Yeah. Where's the? All right, whatever. I was gonna look in my. Oh, here, right here. In my Anki deck, I'll just screenshot it and put it in the notebook.
Okay.
But yeah, I talk about like of course the things that I put in the Anki deck are the things to memorize. So yeah, I put it in.
Is that on the wrong answers deck or the other one you sent me before?
Oh yeah, wrong answers. So basically, yesterday I was starting on a deck where I bring in things from wrong answers and the other thing, and put it all into one.
Mm-hmm.
So, but yeah, so I put it down there, and it should maybe like update at some point.
See.
But yeah, definitely remember that.
Oh, I see it. Yeah. Okay.
Yeah. Myosin, microtubules, and actin microfilaments.
Mhm. Okay.
Um, and so, okay, and also microtubules are the largest, intermediate filaments are in the middle, and microfilaments are, the smallest.
Mhm.
So that can also give, like, you can also learn, like, from that, like, thick, like, thick and thin. I forgot. You know what I'm talking
No, what?
about?
I'm sorry.
Like, the thick part and the thin part of actin and myosin. Oh, anyway.
Oh, the thick and the thin filaments
Yeah.
of in muscle contraction.
Mm-hmm.
Okay. Yeah. Yeah. Yeah.
So if you can remember that microtubules are larger, you will
Mhm.
know that the thick part is made of microtubules.
Okay, and then the thin filaments are the ones that are made of
Yeah.
microfilaments.
Oh, yes.
Yeah?
Mm-hmm.
Yeah.
Yep.
Okay.
Yeah. Good.
All right, next question is amino acids. So
All right.
let's see.
So position 19 of LC20. Okay, so this is a thing that I guess you should know also. Kinase, right? Basically, it takes um it phosphorylates, I believe, through either serine, threonine, or tyrosine.
um, so wait. Let's go back. So it says, so you're saying that I need to know this because they talk about the phosphorylation of LC20.
Yeah, so basically we
Why
need
are they mentioning the position 19? Like,
it just they're just telling you that where it is. I mean, um, but everything that's like that is just so people who take the test are like, oh yeah, this all right. So it has to be at position 19. Okay, and then it says LC20, so 19 and 20. But they want to confuse you and make you think that things like that. But it doesn't matter.
oh,
Mm-hmm.
but wait, it says here in the passage, on the second paragraph, it says, which activates an enzyme that phosphorylates amino acid residue 19
Yep,
of the
that's
actin
talking
light
about the
chain.
same
Okay, I
one.
was like, they never mentioned any residue in the protein, so I was lost on that part.
Oh yeah, they say that in the second paragraph. Phosphorylates,
Okay.
amino acid residue 19 of the myosin light chain LC20.
Mhm.
But anyway, like basically, like whenever like phosphorylation involves a kinase, right?
Mhm.
And kinases, what they phosphorylate, I just think of it as STY. So serine, threonine, or tyrosine. But really, what do those all have in common?
You said PY?
Oh, no. S, T, and Y.
Oh, STNY. So serine, threonine, and
Yeah. How about this? This is a better question. DNA polymerization, right?
mhm.
So what happens there? Like, yeah, like like let's say you have one nucleotide, right? And it's and then there's like like a whatever another nucleotide. How does that polymerize?
I believe that it's through the phosphate group, right? There's a phosphate group that is removed, and then it attaches to one of the carbons in the ring of the sugar.
Yeah. So what exactly does it attach to?
And hydroxyl group,
Exactly.
alcohol group.
Exactly.
Hmm.
So that alcohol group basically is related to phosphorylation, right?
I assume so, yeah.
Okay. And serine, threonine, tyrosine.
They all have an alcohol group.
Yes. So those are the
Okay.
ones that get phosphorylated. So I don't even know what these structures are. I mean, I can guess. But all I'm looking for is the one with the hydroxyl group. So it's going to be that one.
Are these the only amino acids that have a hydroxyl group? Like the only ones? No.
There may be like another, but from the choices, it's the only one. And there may be like one more, but I mean, predominantly it's going to be, you know, the ones like, yeah, it's going to be the ones that have them. Like, yeah, Sarah, like the only ones I can, yeah.
Okay, yeah, it's D. It's D because it's the only one that has the alcohol group.
Yeah.
Oh, wait. I know that everything that you just explained to me, it makes sense, but how did you actually come out with that? So every time phosphorylation happens,
Yeah.
it has to happen through an alcohol group?
Yeah, because, like, so yeah, when you think about content, instead of thinking, because I know I said it happens with these amino acids, and I was going to explain that, but that would be too much just telling you. So I asked you about it, what happens when it happens with DNA, and you were able to correctly get that answer, right?
Mm-hmm.
And that's how you know.
Okay.
And yeah, and I'll tell you like exactly what happens. The OH group acts as a nucleophile.
Mm-hmm.
And it attacks the phosphate or the phosphorus atom in a phosphate and creates a bond with it. So that's why, you know, it in between nucleotides they're phosphodiester bonds.
Yeah.
Yeah. So, you know, try to see everything as like through like a very interdisciplinary approach, and make as many connections as possible. That way, like you have like just multiple ways of answering a question that will get you the right answer, which just raises the probability of it being right. So, and if you didn't know the content, you can like you didn't know that it was with serine, threonine, and tyrosine, but you know that when like DNA gets phosphorylated, right? You know, it's an OH group, and looking at these, we're gonna look at what's different. So even if you didn't know that, I would choose D. Because A, I just get something, you know, I guess it's glycine. Then B, I get something. Maybe it's like isoleucine or something. It doesn't matter. B and C, like they just, what's the difference between them? There isn't really like any difference. But D has an OH group, and whenever you see that OH group, that's why you see the OH group so much.
Yeah.
But yeah, it's a nucleophile.
Okay.
So.
Got it.
Yeah, and yeah, try to connect as many things as possible. I see OH, I think nucleophile, which is a Lewis base, which means that its electron pair is going to nucleophilically attack something that is, you know, electrophilic, roughly.
Electrophilic, yeah.
Yeah. So it's D, and I don't even know what it is, but it must be either serine, threonine, or tyrosine. Well, I know it's not tyrosine because that has, you know, that benzyl group. So it's either serine or threonine. It says it's serine, but yeah, we know. So amino acids right there. Right?
We didn't need to know any specific.
Yeah.
Okay.
So, yeah, any questions?
Nope.
There. Let's jump to number 8.
Okay. So which amino acid is least likely found in one of the transmembrane domains of the GPCR 43?
Yeah, and we don't need to know anything in the passage.
Oh, really?
Mm-hmm.
Oh, it's just going to be a nonpolar.
Mm-hmm.
This is why you need to know the nonpolars. I guess it's glycine.
It says least likely.
Oh, least likely. Sorry. So, actually, asparagine, because asparagine is basic.
Yep, and that's it.
Yeah. Oh my God. All right.
All right, how about
Glycine,
number 13?
PRP is tryptophan. Okay, phenylalanine. Okay.
Yeah, basically they're all going to be... All right, whatever. But, all right, try 13.
13?
Mm-hmm.
Okay. Unbranched alkyl side chain. Okay, I know isoleucine has a branched side chain.
Yeah, what can you tell me about alanine, though?
I don't know much about alanine. I only know it's non-polar. Do you need to know which ones are polar and non-polar?
I mean, like, not really, no. But, like, I guess I start from, like, the simplest, and I know, like, maybe, like, one or two of those. Well, I know the two simplest ones, and then I know, like, the basic ones, the acidic ones, and then the ones that get phosphorylated, aka the ones with the hydroxyl group. And then there's one that is a secondary amine. So that's how I think of it. And that's like an active approach. Instead of just memorizing them all without rhyme or reason, I separate them into those categories. So, like someone could memorize the structure. So if I asked you which amino acid, has a secondary amine, people who memorize them all, they can know the structure of them all, but they're not going to be able to answer that because they just
They kind of like, so it's basically when you memorize something with a particular set of conditions, and then they switch it around, and you're like, I have no idea.
Yeah, that's why rote memorization never works.
Yeah.
So what? Yeah. So now, what do you think is the one with a secondary amine?
I don't know. Wait. No, I don't know.
All right, it's okay that you don't know. Remember, tolerance of uncertainty. So think about it. In order for it to be secondary amine, right? It has that amine group has to be attached at both ends, right?
To two groups, yeah.
And this is one amino acid. It's not attached to another one. So it must be what?
A ring.
Yes.
Is it proline?
Yeah.
Proline has an amino?
Yeah.
It's proline?
Yeah. And you see how you got that?
Yeah, I know proline is special in many things, like also causing the kinks on the beta sheets and all that stuff.
Yeah. But you see how that all just, you don't need to know that it's going to cause kinks because you know that if you know that. So like, it's great. So that's how I'm, like, asking you questions, like, in these, like, kind of small steps because like I asked, like, okay, in order for it to be secondary it must be what, and then you're like, so in your head you could have every single amino acid. There's more than 20. There's 20 that we essentially use in our human body, but there's billions, right? You could be like a computer or something, or like a textbook, but it's about like recognition, not recall. I'm sure you've heard that before. So
yeah.
So and this lets you kind of do like a search function in your head, And so you're able to deduce that it has a ring and then correctly say that it's proline. And if it has a ring, right, it's going to get in the way of things. So it'll get in the way of making, a beta pleated sheet keep going in one direction. So that's where the turns happen.
Wait, there's many others that have rings, and apparently proline is the only one that causes the kinks, right?
Yeah, but do those rings have whatever? Those rings are whatever, like a benzene or something like that. Tryptophan is like a double ring structure. But yeah, you're just looking for the that secondary amine.
Okay.
Because that is what's bound. All right, let me put it in a better way. Proline is a secondary amine because the secondary amine in it is the N terminal, whereas all the other ones, it's the side chain which could have a ring.
Oh, the secondary amine is the actual amine of the N terminal?
Mm-hmm. Yeah. So other things would have it as like a side group or a side chain or whatever, but it'll still have that primary amine though.
Okay, hold on. I just want to see proline.
Mm-hmm.
Okay. Yeah. Okay. So, we haven't answered the question.
Oh, okay. So they're asking for an unbranched alkyl side chain, and okay, well, I mean, I don't want to say that because I know this one, because I know the two simplest ones. But you probably
isoleucine and leucine, they're pretty much they're isomers, I guess.
Yep.
So they're both going to be branched, right?
Mm-hmm.
So then that's out.
Mm-hmm.
And then valine and alanine, I don't know the difference between both of them.
Mm-hmm. So on any list of amino acids, what's usually the first one that they list?
Alanine.
That's like the second one that they list, but
glycine is the simplest one.
Yeah,
and then it comes alanine, valine.
All right, so glycine is just an H.
Mm-hmm.
And then what's less simple than just an H, but not what's like the next step? Like
you're adding a C carbon.
Exactly.
Or like a CH something.
CH3.
Yeah.
Like just a methyl group.
Right.
Yeah.
So you have the hydrogen, you have the methyl group, and then you add the CH2CH3, which is going to be valine.
Yeah. So the way I see it is like simplest amino acid just has an H. Then it's like, okay, let's try to be a little bit more complex here. Let's add a methyl group. So you do that. And then after that, it's like, all right, maybe we can add a methyl group that has another one, like an ethyl group or something, right?
Mm-hmm.
And then let's have, which I assume is leucine or whatever. And then isoleucine would be like, let's take that that ethyl group and let's move one of them to be like, on like this carbon. So it looks like kind of like an isopropyl kind of thing. But yes, this is going to basically be alanine, and that's just the one that has just one amino. I'm sorry, just one methyl on it.
Right. Hold on. And valine has two methyls and a CH. Okay. Okay.
Yes, and I'll okay, so that's that one. So yeah. See, I'm just going through like the amino acid
Yeah.
kind of questions. All right. How about 21?
Okay. 19. Okay. Which
So
amino acid?
without looking at anything, like, tell me about these amino acids. We're gonna do like like what's different.
Okay, so asparagine is basic, glutamate is acidic, lysine is basic as well, and valine is, I guess, non-polar.
Just neutral is good enough.
Yeah, neutral. Yeah.
So be careful with asparaginine or whatever.
Ooh,
Mm.
yeah, they're both acidic.
No. Oh, so okay. There's aspartic acid and there's asparaginine, just like there's glutamine and glutamic acid, right?
See, that is very so. Okay, so because when I read it on the book, it said so it's glutamate and glutamic acid, and there is aspartate and aspartic acid, right?
Oh
Which
yes,
are
yeah.
the
I meant oh, I forgot.
yeah. So aspartate, and glutamate are the acidic ones, and then asparagine, or I don't know how to pronounce that thing. It's
Yeah.
the basic one.
Oh.
Okay.
So it it's not the basic one. It just is essentially what happens is, so okay, let's say you have aspartic acid and you replace that carboxylic acid group with an amine, and then you get
Okay.
asparaginine.
No, really?
Yeah. And it can help, and if it ends in ine, that's gonna kind of, well, actually, that's not gonna really help you. But the glutamate and glutamic acid, like acids, will be like when they lose a proton, they'll be like the whatever the salt version of it, like the 8. It'll end in 8 or whatever.
Yeah.
So yes, so we could treat those as the same. But yeah, asparaginine is it's like it's basically aspartic acid with a amine group instead. And then the other one.
Is there any basic amino acid that comes from glutamic acid? No.
No. But there's a amine version of that too, which is glutamine.
What?
Yeah.
Okay, wait. This is like
Mm-hmm.
okay. So glutamate and glutamic. Okay, glutamine. Glutamine is a basic amino acid.
No.
No.
It just you take that carboxylic acid out and you replace it with an amine, so it's glutamine.
But it's
So this is
it's polar. It's polar in charge. Okay.
well, we don't have to worry about polar or non-polar.
Sorry, I just wanted to know where it belongs to.
Yeah, basically, if you see like a benzene group or something that's like a ring, right? It has a lot of symmetry in it, so that's going to be non-polar, right? And everything else is going to be polar. It's better to let's how should I put it? It's better to know the things that are different, because everything else will just be the same, instead of knowing everything that's the same and then going from there. So like again, like even though I never just sat and memorized the amino acids. However, I take note of the ones that are different and how they're different, and that kind of lends me some knowledge, but it's through deduction.
Right.
Because like if you just memorized like glutamine looks like this and it's non-polar, blah blah blah, I see it as glutamine and glutamate. There's definitely a connection there, and the connection is that it's just you take that carboxylic acid out and you put a amine group there. Same thing with asparagine.
Well, you only take the alcohol, no?
Well,
From the carboxylic
if you leave
acid?
the carboxylic acid, it would still be an acid, so it would be called an acid.
Oh wait, so like when you have glutamic acid, because the only difference is that it still has that carbonyl though, but then instead of having the OH of the carboxylic acid, which is in glutamic acid, it has the amine group.
Mm-hmm.
So you're basically taking just the OH, right?
Yeah, but okay. So I guess when I say you take out the carboxylic acid, I don't mean, like that you're losing that it's being decarboxylated, like you're losing CO2.
Mm-hmm.
So I don't mean that you're losing the carbonyl and the I'm not saying that you're losing that entire thing, but you're losing the part that makes it a carboxylic acid is what I'm saying.
Got it. Okay.
Yeah. And it's what then? It's an amide, I believe, or something.
Yeah, it's an amide, or however you say it. I don't know.
Yeah.
Okay.
Yeah. So that's what you can. So yeah, when you see those, you can think of them, as like that. So basically, okay. So we have asparagine, which is its own thing. Whatever. Let's just try to do it by extreme differences first. So asparagine, it's not going to be charged. Glutamate is acidic, it's going to be charged. Lysine is basic, it's going to be charged, in the opposite direction. Valine, not going to be charged. So basically, B and C are saying the opposite things, and A and D are pretty much saying the same thing. I mean, yeah, to me there isn't much of a difference because they're not charged. So now we just have to see if it's going to be, the one that's positively charged or the one that's negatively charged. So I'm going to look at position 64, and I see it, D64.
Wait, you're too fast. Let me look for 64. Okay, D64. So the CCD contains 3 highly conserved residues. Okay.
All we need to know is a D64, and D is what? So I have a way of remembering it.
D is aspartic
acid. Yeah, I think of it as aspar dick with a D acid.
Yeah. That's why I remember too. And then glutamate I actually remember because it ends in E, so yeah.
Oh yeah, isn't glutamate Q?
Glutamate is E.
Okay. Oh yeah. All right. Yeah. Oh, maybe I think of glutamine. Yeah, I'm thinking of whatever. Doesn't matter. But okay, so
glutamine is Q, yeah.
Oh yeah, so I think of it as cutamine.
Oh, okay, got it.
But okay, so D64. So what? It's a acidic one, right? So if we change it, what's gonna have the least effect in what's gonna basically be the same thing? How can you replace it and not have it really change things? And how do we keep it functional?
Right. So if glutamate is actually acidic, it is charged. Yep. So how we do not or the way that we would least affect it would be adding something else that is charged.
That isn't in the so it's already aspartic acid, right? So it's negatively charged. So we want to replace it with something that's also negatively charged, right? Yep.
Yeah.
So glutamate, right?
Well, wait. We said that we were actually ruling out B and C.
Oh, no, I was saying that we're ruling out A and D because those don't have charges.
Oh, got it.
And then,
Okay, and then we were between B and C.
yeah,
Okay.
if the question was most likely to affect it, it would be C.
Mhm. And really, okay, so if we only, like, before having all of this discussion about the amino acids, if I would only look at D64 and know that, so D is glutamic acid, right?
Mm-hmm.
And then glutamate is also the same glutamic acid without the H. So it's a salt of glutamic acid.
Yeah, treat the salt and the acid as the same. Because it's going to be in water, right? So it's going to be basically, going back and forth between, the salt and like getting the H, like removing the proton, getting the proton, right? Mm-hmm. In fact, it's going to be, because whatever, Zwitterion and stuff. But yeah, treat them as the same.
Okay.
Yeah.
I just don't understand why they're saying like amino acid substitution, knowing that it's the same amino acid though.
It's not the same because it's supposed to be aspartic acid or aspartate.
Oh yeah. Oh my god, yes.
Yeah.
I'm sorry. Yeah, D is aspartic acid, and then it's being changed for glutamate, which is glutamic acid.
Mm-hmm.
Okay.
Yeah, I think this is whatever. So missense mutation or something. Missense mutation. Change of a single causes a different amino acid, and it may not have no effect. Oh, okay. Anyway, because they when they ask questions about mutations, right? Mm-hmm. And a mutation could just produce an amino acid that serves the same function.
Mm-hmm.
So that is another thing that I'm connecting this to.
I will most likely ask a question about that.
Oh yeah, what's up?
That they will most likely ask a question about that.
Oh yeah, they always do. Yeah, I've seen I've seen a billion of these kind of things, so I kind of like also know like what they'll ask you and what type of thing they So that's why like I know what parts of the content is important.
Okay.
Yeah. But yeah, missense mutation, like let's say there's a mutation and it causes the amino acid to turn from aspartic acid to glutamic acid. Overall, the protein is going to have roughly the same, like similar function, right?
Mm-hmm.
Okay.
Okay. Cool. Not that bad. Do you want me to go to the next one, or you want me to go to another particular one?
Let's go to 35.
35. Okay.
Okay, the amino acid at position 61, so I go to position 61, and it says that it becomes deaminated. It undergoes deamidation.
Okay. I don't know what deamidation is, but I'm assuming it's like the same as deamination, but it's just deamide.
Yeah. So what's the amide? It's kind of like...
It's a carboxylic acid with an NH2.
Yeah. So, yeah. Okay. So, okay. So, let's see. So, basically, rack, right, gets deamidated. There's something, alright. Rack gets deamidated, if that's how you, even say that. At the position 61, right? So we're looking for an answer that is the result of, or rather could be deamide, can have its amide group like removed.
Mm-hmm.
And yeah. But even
already know which one it, I mean, I kind of already know which one it is.
Yeah. Which one?
So glutamine is going to be acidic, so most likely it's not going to have an amine group or amine derivative.
Remember, glutamine different from...
Oh my gosh, yes. Yeah.
It's okay.
Okay, so then I don't know.
So how about this? Let's look at, all right, glutamine and arginine. Okay. And leucine and tyrosine, right?
Mm-hmm.
So tell me about kind of like how we did that last problem. Tell me what's important about these.
Okay, so glutamine, I don't know if it's...
Let's leave glutamine for now.
Let's say it's neutral.
Let's just from B, C, and D, tell me about them.
Okay, so arginine is basic.
Mm-hmm.
And tyrosine and leucine are neutral.
Yeah. So those guys are out.
Mm-hmm.
Okay, now... Alright, so glutamine is just like asparagine. And remember how we talked about that, right?
Yeah, we just talked about it.
I'm
sorry.
No. Don't be sorry. No. So okay. Now okay. So okay, they're saying that this thing gets deamidated, right? So its amide group is removed.
Mm-hmm.
And which one has an amide group?
Glutamine.
Yep. So that's it.
You know what? I was actually going to go by arginine just because I forgot what you told me about glutamine.
Mm-hmm.
And I was like, arginine is basic, so most likely it's going to have an amine group or some sort of amine derivative just because it's basic.
Uh-huh. Yeah.
But then I remember what you said about glutamine. Okay. So
yeah, and then
important.
and you remember like amides are different from amines, right?
Right.
Mm-hmm.
Okay. I want to do actually the one that was before the number 33.
Oh, okay. Yeah, I was just going through, the ones with amino acids.
Oh, no. Oh, yeah, it's fine. I just saw it while I was
we could
trying
do that.
to get to 35.
So let's see. So, okay. Km is the same, so it can't be... Alright, let me just... Okay, so blah, blah, blah.
You're like, blah, 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 who cares? Like...
That's, usually how it... Yeah, okay. So, it's not going to... If the Km is the same, what type of inhibition can it not be?
If the Km is the same, it's not going to be competitive.
Okay. Now, which of these choices is talking about competitive inhibition?
D.
Yeah, so it's not going to be D, right?
Mm-hmm.
Okay. All right. Now,
wait, how do you know that the Km is the same, though?
Because they're all... So all of these lines have the same,
they're converging. Yeah. I'm sorry. I was looking at the B maps. Yeah, so they're actually all stemming from the same point. That's true.
Yes. Okay, so that's how you like. Basically, you're looking for those two things. Like you're looking for whether or not that because that determines Km, right?
Mm-hmm.
And then the other thing that could change is like Vmax, right? And that's gonna basically affect like the y-intercept of it.
Mm-hmm.
And so that arrow that with that I means that as we increase whatever inhibitor concentration, we go from that line. What like the slope is basically increasing.
Right. The slope is increasing, but then because it's the inverse, then Vmax is decreasing.
Yes.
Okay. So that is not really important, I guess. It doesn't take me to any answer anyway.
Well, okay, we can look. All right, so if we look at the choices, like irreversibly binds to the enzyme, can only bind after it's already after it. So B is saying that it can only bind to the enzyme substrate complex, and then C is saying that it binds the enzyme and the enzyme. Oh, that's funny. I can hear myself.
Hello?
Yeah.
Oh, okay, yeah. Sorry, can you repeat that again? Because my AirPods are garbage.
Sure. So yeah, in terms of inhibition, like so with competitive, KM changes, Vmax does not. With I always mix up uncompetitive and non-competitive, but I know that for one of them, like KM stays the same and Vmax changes, and for the other one, they both change.
Yeah, so for uncompetitive, both of them change.
Okay, so then it's gonna have to be non-competitive.
Well, but KM is staying the same.
Mm-hmm.
So it's definitely not it's gonna be non-competitive, yeah.
Okay. So yeah, so VM is changing. So is that something that only binds when so B is saying it only binds to the enzyme after the enzyme has already bound substrate. It should say bound to a substrate. And then C is saying it binds to enzyme and enzyme substrate complex with the same affinity. So what do you think? Which one is non-competitive? Because I honestly don't remember. But I don't
non-competitive is whenever it's like allosteric inhibition, right? So basically, it would bind the enzyme before like the substrate actually binds to it.
Really?
And that's why the Vmax goes down.
Okay, I think because I remember something with it binds. I remember three things that could happen. One, it binds at the same active site, which is competitive. Another one, it binds at a completely different site.
Right.
And then another one where it binds to the enzyme somehow, and it also binds to the enzyme that's already bound to the substrate.
So that would be uncompetitive because I know that the one that binds to the enzyme at another different site, it's non-competitive or allosteric inhibition.
Okay. So,
but then if you read like B and C are pretty much saying the same thing. Like I was trying to look for an answer choice that said.
Well, C is saying it could bind to the enzyme as well, though.
Bind the enzyme and the enzyme substrate complex with the same affinity. Okay.
And B is saying it can only bind to the enzyme substrate complex.
Okay, so then it will be C.
Yeah, that's what I would say. I honestly don't remember the different types. Okay. But through process of elimination, we're able to get C, and it seems like it's non-competitive, and then they bind the enzyme substrate complex and the enzyme with the same affinity. Okay. So, that one makes sense.
Yep.
And any questions about Lineweaver-Burke? Like why it exists?
Uh, I kind of read why it exists. I don't know why they have to complicate stuff, like
Because
in the
it looks
sense of
very
like
complicated.
reversing
Yeah.
it with inverses. I know it's because it's an asymptotic function or something like that. So
Yeah, like Michaelis Menten is asymptotic. Um, so it's a way for them to make it a straight line.
mm-hmm.
Um, yeah, it's weird. I never liked it.
Oh my God, thank you. I hate it. Like I'm like I actually like more
Mhm.
the Michaelis Menten like
Mhm.
the graph or how it actually works, and then they're like, surprise, now we have inverse. Like I don't work that well with
Mhm.
inverse like
Yeah.
graphs and stuff like that.
Yeah, because
I don't
it's hard because if km goes up, like the number
But it goes
d.
up in magnitude though. So that's
Mhm.
the confusing part.
Yeah. So, so like that means that it'll move like to the right because yeah, it's weird. But yeah, I was looking at the other one that talked about um oh yeah, 42.
Okay.
Oh yeah, this is good. So it says what is at residue 119, and I see that it's sarin normally, right?
Mm, you're so quick. So I was just reading the question. PK phosphorylates CREV 327 at serine 119. Okay. And then it's saying which of the following residues was most likely chosen as the substitute. Okay. So can I
Oh, okay. You know
say
what?
that the
Wait, it says
only
that they're
thing
making.
would be
Oh, they're
oh
they're making a variant. All right. So okay. Uh, whatever. We know that it's sarin at 119 and that it gets phosphorylated, right? And then they made variants of it that cannot be phosphorylated. So we're looking for things that can't be um, phosphorylated. And we know that sarin can, we know threonine can, and we know tyrosine can.
mm-hmm.
And I guess this is a good question that you were asking. Like, maybe there's another one that will have an OH group, and
Mm-hmm.
yeah, that could also happen. I suppose it's not usually the case, but like if I had to choose between glutamate and alanine, like I would know which one definitely can't be phosphorylated, right?
Alanine.
Mm-hmm.
Okay, but then you just went too quick. So,
Mm-hmm.
whoa. All right. So, PK phosphorylates CREB 327 at serine 119, and then the variants of wild type cannot be phosphorylated. Okay. Okay, that's what we needed to know. Yeah.
Mm-hmm. Yeah. So I
Like
looked
you
at
read, okay,
yeah
so on top of the fact that you're like talking to me
yeah.
and paying attention to what I'm saying, you're also reading the passage at the same time, and you just came up with the answer like that. That's
Well,
amazing.
I'm just looking at the question, and then I'm looking for 119, right?
Mm-hmm. But then we needed to know what the variant did because they were mentioning their the new
Yeah. Then I looked
yeah,
at
the
it
wild type.
again because at first I was going to be like, oh, it's going to be another thing that acts like sarin, and then I see
Exactly, that's what I was looking forward to.
yeah, and then I see tyrosine and threonine, which both act like sarin, so those can't be it.
Oh,
So then I
yeah.
looked at the question again, and it says it's a variant, and then I looked at the part where it says variants were that cannot be phosphorylated.
Mm-hmm.
So it's like a need to know kind of thing, right?
Yeah.
That because like again, like in that slide, like our working memory can only store 7 ± 2 bits of information per time. Reading like the entire passage like is too much at once. We're not going to be able to retain that at all. Like yeah. So I keep it like like it's like I have like a low information diet almost. Like Just I look at what I need to look at and just take it a question at a time. I don't even look at that gel. I don't want to.
I was gonna start looking at the gel because
Yeah.
I was like, okay,
Don't-
there has to be something in that gel that tells me whatever
Yeah.
is different from the mutated protein or whatever.
Yeah. Only if they ask you. All right? So, no, like, when I see that, I'm just like, nope. And, I go through the questions, and if I need to, then I'll look at it.
Okay.
But, yeah. Think about, what things are, whatever. Like, does this need to know kind of thing kind of make sense?
Yeah. No, it does make a lot of sense.
Okay. Good. All right. How about 49?
Okay.
So generally speaking, so I knew from looking at this that it's an amino acid question, and it's like a amino acid substitution. Like you'll have the letter for the amino acid, a number for the location of it, and the letter after it is what you change it to.
Okay.
Okay, and before looking at anything, I'm going to look at which things are different, which things are not. Right away, I see
the answer choices.
Yeah. So
Okay.
For instance, right away I see valine turning into leucine. That's out. Who cares? Like no one cares about those amino acids. We only care about you if you have if you're if you're whatever basic, acidic, can get phosphorylated. I don't care. Valine and leucine, who cares about them?
Okay.
No one cares. No one in the
okay.
world cares about you guys. So you're out. You're never gonna be the answer, unless it's like
They're never gonna,
unless
you're never gonna be the answer to anything.
unless it's like it could be the answer if they say something whatever is lost or something. But anyway, then I see Q to N. That's glutamine to.
To asparagine.
Okay. So yeah, I didn't have that remembered. You do, so that's good. But I would have known that like Q is not charged and N is not charged because I know the ones that are charged. So I'm gonna say that's out. I mean, I could be wrong because I haven't looked at it yet. So, but if I look at the A and B, we have some charged stuff, right? So K is lysine. R is arginine. So it maintains its basicity.
Yeah, both of them are basic.
Yeah, and then what about B? I'll be right back.
R is arginine, and then D is glutamate. Sorry, D is aspartic acid. Man, I keep confusing them. So you're going from basic to acidic.
Yeah. So right away, that's what I would choose.
Oh my god, you didn't even look at the positions in the passage.
Yeah.
Is that the right answer?
Yeah.
Oh my god. Wow. Okay. Well, so you said that you didn't remember what N was.
Yeah. But I know that it's not charged, and I know that Q isn't charged because I know the ones that are charged.
Right.
So it's
well,
yeah.
But that's the thing though, like in the list it says like, well, I know you don't want to memorize the list, but it says like polar uncharged and then polar charged.
Yeah. So they're both polar, so who cares about that part? The thing that is different is what matters. Right?
But they're charged. So what does it mean if they're charged?
Oh, you know what that means.
Well, that they have a charge, but so they would technically have a charge, no?
What?
If they're polar, hold on, let me look for them.
So
If they're
if you're comparing polar uncharged to polar charged, who cares about the fact that they're polar? Because that's the same for both of them. The difference is that there's uncharged and charged.
okay,
Does that make sense?
yeah,
Like, let me put it this way. I'm going to tell you to remember all of, in the alphabet, all the consonants and all of the vowels. And if you just remember the vowels, which there are just what, like 4 or 5 of,
yeah, 5.
then you know all the consonants. Because they're the ones that aren't those 5.
And you know whatever doesn't belong to any of those 2, then who cares?
Yeah, like, so instead of remembering every single thing, does that make sense? Like,
Yeah.
yeah. Let's see if I could find another one. Okay. How about 56?
Okay.
Now, what are your thoughts about transmembrane?
So it's gotta be nonpolar.
Okay. So, let's first of all look and see if we can see anything that is charged in any of the other choices.
Yeah, so B has lysine.
Yep.
C has lysine and arginine.
Yep.
D has D,
Yeah. So D doesn't have anything that's charged.
and A doesn't have anything that's charged either.
Yeah. So start off like that way. So B and C are out.
Mm-hmm.
All right. Now, let's go from there. Now, we know that serine has an OH group, right?
Yes.
And that makes it polar, doesn't it?
But so it's STY, so that's 309 also has.
Yeah, and tyrosine?
Mm-hmm.
Yeah.
So all of those are polar.
Yeah. So, like, yeah, instead of, like,